Hi everyone, it's Mr. Vallejo. Welcome to biology class. Today I'm going to talk about biochemistry. So let's go ahead and share that screen with you and we'll take a look at the PowerPoint which you can access in Canvas or Schoology or Ingenuity or whatever learning management system that we're using this semester. All right, so let's get this show started. Looks like this. And we're going to go back and show you the right screen. So as we take a look at biochemistry, what I want to tell you for the second midterm exam is that the first 17 slides are going to be vitally important for you. That's because the questions, 20 questions on the next exam are directly from the first 17 slides. Here's the first one. Um, the uh, the questions will be based on the header up here. It says here the gene is a functional unit of heredity, but I had to change that into a multiple choice question. So if you'll do uh, if we take a look at these headers and learn them well, you will be pleasantly surprised when you do very well on the 20 questions that are going to be on the test that are based on these headers. So it says here the gene is a functional unit of heredity. So as we pass along characteristics from parents to their offspring, those characteristics are passed along by means of the gene. Remember, the gene is simply a part of the DNA. Stretches of DNA that program the amino acid sequences of proteins. The proteins are the gene products. So what the DNA tells us is it tells us what proteins that we need to make. So the gene is the functional unit of heredity. Genes have DNA codes that act as blueprints for the making of proteins. That's why I have in the bottom left corner, there's a blueprint of a plane. And so you can see here, it's an image we've seen before from previous lectures and previous talks. It's the double helix of DNA, deoxyribonucleic acid. And, and so the genes are made up of DNA sequences. And so those DNA sequences uh, have the directions on which proteins to make. Now, these proteins then are the final product of the gene expression. Sometimes it's not quite a final product, it's called a polypeptide. This is a polypeptide. It is part of a protein, but for this particular um, protein, this particular protein is made of 4 parts, 4 polypeptides, and when you stick them together, you get uh, a transthyretin protein. Okay. The human body has many different cell types. Nearly all cell types in the same individual have the same DNA sequences. It's like the library. And the library has all these different books, but you probably don't need all those books if you're trying to fix your 1971 Impala. You got to get the right book, and that book, that Chilton's repair manual, will have all the directions on how to fix whatever you're trying to fix. But if you happen to get the book on Baryshnikov and ballet, that's not going to help you all that much. But it's there, and so all the cells in your body, just about, we'll see some exceptions, and we have seen some exceptions as we study mitosis and meiosis. All those cells in the body have the same DNA sequences. So to help illustrate this, let's take and consider an eye cell and a skin cell. Well, the eye cell and the skin cell, they're very different, but they have the same information. So here's a question for you. Which is true for an eye cell and a skin cell? Do they have the same genes and have different active proteins? Do they have different genes with different active proteins, or do they have the same genes and the same active proteins? We'll give you a second. Okay, what do you think? If you said A, same genes, but they have different active proteins, then you are correct. The term genome means all the cell's DNA. And so when you're taking a look at the 46 chromosomes and all the information that those 46 chromosomes have, that's called your genome. The nucleus houses most of the cell's DNA. Both the mitochondria and chloroplasts have a small amount of DNA. We covered that in the cytology lecture. It is thought by many that chloroplasts and mitochondria were actually bacteria on their own, which were engulfed by larger bacteria, and because of the symbiosis of the mitosis and chloroplast living within that larger bacterium, that's how eukaryotic organisms, or at least eukaryotic cells, came about. So the nucleus is right here, and it houses most of the cell's DNA. Here is a karyotype, as we've learned about before. Hopefully you'll know that this right here tells us that this is a karyotype of a male. But if you look at all of these, there's 23 pairs altogether. 23 times 2 is, human cells have 46 chromosomes. And then each of these 46 chromosomes are paired up. So these are called homologous pairs or homologs of each other. And so we have 46 chromosomes that are divided up into 23 pairs. Those first 22 pairs are the same in males and females. They're called autosomal chromosomes. But that 23rd pair you can see is different. Take a look at that one compared to that one. And just so we can see it bigger, this 23rd pair is called sex chromosome. This is a female's sex chromosomes, and this is a photo of a male's sex chromosomes. This is called XX, and this one's called XY. We'll learn that the Y has actually little genetic information. In humans, the Y chromosome is absent in males. So in the absence of a Y, you're a female. If you have a Y, you're a male. When I was sitting where you are, this number was 100, 120, 150. They weren't quite sure. But now we think that there's only about 20 or 30,000 different genes in the human genome. Only 30,000 different directions that we need in order to make a you. Now, those chromosomes are mostly empty space. Those DNA strands is mostly blank. It doesn't say anything. Most of the DNA in the human genome does not code for proteins. If you take a look at these two pictures, here's one, here's another one. This most of the empty space is called heterochromatin. Euchromatin is where it all happens. Remember, EU means true. Chromatin is a DNA protein complex. So this is the good gene. Over here, heterochromatin, that means different gene. So I liken it to the Highway 5 going up from where we live up through the Central Valley. I just drove that last week. Oh my goodness, it was horrible. Okay, so you go down the Grapevine, which is a really big hill, and you get down to the Central Valley, you start driving, and there's no cities. There's nothing until you get to the first major, it's not even major, the first spot, first stop. It's called Buttonwillow. You can get a taco there, go to the bathroom, hop back in, and you start driving, and for the next 30, 40, I don't know, 50 miles, it's the same thing, the same hill, the same power line, the same stinky cow. And you just keep going, and there's nothing until you hit the next thing called Lost Hills, and you can get some gas if you need it. You can get another taco, you get a bag of chips, and a soda, hop back in your car and do it all over again. So euchromatin and heterochromatin, most of the DNA, just like I5, is empty space. But every once in a while you get something good. That's a euchromatin. That's a taco joint. Now, I don't usually have you guys memorize numbers, but here's some numbers to know. There are 3 billion base pairs in the human genome. Of those 3 billion base pairs, 99.9% are exactly the same as that 0.1%. It makes everybody different look. Can you find anyone exactly the same here? I mean, there's 7 billion people, 7.5 billion people on Earth, and no two are exactly the same. That is well designed. Yeah, on this one here, it says some genetic diseases can be predicted based on the presence of a particular gene sequence. Other genetic diseases are too complex to be predicted at this time. So, just because you carry the gene for cancer doesn't mean you're going to get cancer. Now, your chances are higher, but you don't have to get it. There's some things that happen that could trigger turning on this gene or leaving this gene off. So, we need to know more about this. Right. So if you take a look at the slides up to this point, there's a lot of information, but be sure to concentrate on the top headers, the top of the screen. Those will help you open the door to 20 of the 100 multiple choice questions on the next test. Now, for the next part of this lecture, I would encourage you not to take notes because it might be frustrating, but what you need to know is generally this information, and I will go through it pretty quickly. You also have the PowerPoint at your disposal, so you can take a look at that and uh, and go through it at your own pace, or you can take even just the recording and pause and rewind and such. Okay, so the central dogma of DNA. Dogma is your basic beliefs. Like the Catholic Church has a basic belief about the Immaculate Conception and Mary is the Mother of God. Okay, so that's what you believe if you're a Catholic. That's your dogma. So the central dogma of DNA is that there's DNA, there's RNA, and there's proteins. So we can't see DNA, but we believe that this is the way it works. That DNA is stuck in the nucleus. We have to make a copy of it, and that copy of it goes out called RNA, and then we can read that or translate the message that's on the RNA into the final gene product until we get the protein that we're interested in. Just like when you go to college, they don't want to see all your work, they don't want to see all your essays, they don't want to see all your scantrons. They just want to see one final piece of paper. So when you left high school, or if you're a transfer student, you're trying to get into Big U, well, you need a transcript of your grades, just like that. We don't need the whole DNA. We just need the little piece that means something. That's a transcript. So DNA transcription gives us the RNA. Now, the RNA, it's in a foreign language, it seems like. So that RNA has to be translated, and then the ribosome can make a protein out of that. Also, DNA needs to be replicated and you need to pass the information on to the next cell. And so DNA replication is a process that we're also familiar with and also makes up part of the central dogma of DNA. Now remember the structure of DNA, it's the double helix. It's like a ladder that's twisted, it's double-stranded. And it's made up of parts called nucleotides, but the nucleotide is made up of 3 subparts itself. It's shown here, the phosphate, the sugar, and the nitrogen base. Nucleotides form the backbone of the DNA structure. There's the sugar, there's the phosphate group, and there's the nitrogen base. You can see right there. It's those nitrogen bases which make up the DNA code, as we'll see in a little bit. Now, the DNA is a nucleus, but remember we have the cytoplasm nucleus, and the DNA is stuck in that nucleus. It's protected in there, and you can consider it to be locked in the vault. But all those proteins that we're interested are made outside the nucleus, the cytoplasm. Now these proteins are chains of amino acids, so what we have to do is we have to take a look at the transcript and translate it and see what the sequence of amino acids are. So these proteins are made by a protein factory, a ribosome, in the cytoplasm. So the ribosome, in our analogy, is the protein factory. Now, how is this information passed on? Well, the DNA is too big and it's stuck in there. It's protected in the nucleus. So what you need to do is you need to make a copy of the message of the part of the DNA that you actually need. You don't need a copy of the whole thing. You just need a copy of the little piece that tells you how to make the protein. This is copied on a molecule called messenger RNA. Messenger RNA is so small compared to DNA, it's able to get out. Once it gets out, the ribosomes can read those blueprints and then build those proteins. So, in other words, you got the DNA, which is all the directions. The DNA is copied onto the messenger RNA. The messenger RNA leaves the nucleus, and then those copies are read by the ribosomes. The ribosomes then take the amino acids, put them in the right order, connect them, and then those proteins lead to characteristics or traits. Now, the differences between DNA and RNA, there's your DNA, there's your RNA. DNA is on the left, RNA is on the right. You can see the name is slightly different. That there is a part of the sugar that is missing oxygen on the DNA, so we call it deoxyribose. So we have the deoxyribonucleic acid and the ribonucleic acid. Nitrogen bases in DNA are A, T, C, and G. They pair up this way. A pairs with T, C pairs with G. And that's a base pairing rule. Now, in RNA, it's slightly different. There are no T's. There's only these things called U's for uracil. And so wherever you expect a T, you've got a U instead. So that's uracil. DNA is double-stranded, much larger. RNA is much smaller. Now, remember, our mRNA is the transcript. mRNA is a copy. It's just a copy of the small piece of DNA. So that copy of the small piece of DNA, or the copy, which is the small piece of mRNA, is able to leave the nucleus. This happens because of an enzyme called RNA polymerase. And if we look at it, remember ASE means it's an enzyme. It's going to make a large molecule or polymer, and that large molecule is called RNA. So this makes sense. It's RNA polymerase, mRNA, or RNA polymerase. So RNA polymerase is what is the enzyme that is involved in transcription. So matching base pairs of DNA and RNA. So what happens is the DNA unwinds and unzips, and then what happens is the RNA polymerase reads the open DNA strand and puts those RNAs in place. And so you can see it happens like this in the mRNA. Well, after the mRNA goes out to the cytoplasm, then what's going to happen is the ribosome is going to read it, and the ribosome puts in the correct place the amino acid, which again, a protein is a long sequence of amino acids. And those amino acids then, put in the right order and in the right composition, will result in the proper protein. Those proteins then lead to trait characteristics. Now, the RNA, before it leaves the nucleus, it has to get what we call processed. So the mRNA is processed and it leaves the nucleus. Then it goes to the ribosomes, which are the protein factories. And then those proteins are built from the instructions on the mRNA, which are called what's on the DNA. There's the sequence of amino acids right there. Now, what is this processing about? Well, what happens is just like when you dress up for the prom. Remember the prom? When I went to the prom, you got a tuxedo. My friend Mike got a hat and a cane. Woo! Well, you get all dressed up. Well, just like that, going to the prom, mRNA, as they leave the nucleus, they need to get one of those fancy caps. The cap is called the Pribno box, and we're going to put that, it's a 200 base pair sequence that sits in the very front over here. And then you got to get a tail on your tux, and this tail over here is made up of A's. You actually call this the poly A tail. Before the mRNA leaves, there's parts that don't mean anything. So we cut those parts out. Those are called introns. And then once you cut out these parts that don't mean anything, you can stick the meaningful parts together. And so that mRNA then ready to go out and go to the ribosome. The ribosome reads the messenger RNA, puts the amino acids in the right order, making the protein, and the protein will lead to characteristics or traits. But the question is, how does mRNA code for what it is that as the ribosome reads across each of these nitrogen bases and A, U, G, C, G, U. As you put them together, each sequence of 3 means an amino acid. So when you look at it, what's happening is that you got a triplet code. AUG means methionine. CGU means arginine. There's a code which is redundant in that there's only like 20 different amino acids, but there's 64 different combinations of 3 different letters this way. So as you can see, we have these triplet codes, and these triplet codes are called codons. And there they are, they're the different combinations, and you can see that sometimes it didn't matter what the last letter is. CU what? Well, those all mean leucine. But there's only one start, and that's AUG, and there's 3 stops. And the 3 stop codons are right there. So the start codon is AUG, but the stop codons are UAA, UAG, and UG. Now, how are the codons matched to the amino acids? Well, there's another type of RNA, tRNA. The T stands for transfer, and those transfer RNAs correct amino acid to the location. Put those in a part of the ribosome. There's the uh, amino site, and there's the protein site or peptide site, and then there's the E to N site, APE, and those are all parts of the ribosomes. And so what happens is the transfer RNA brings the correct amino acid to the right place according to the anticodon, which matches the codon. So the codon to transfer RNA is called the anticodon. Clever, it matches the codon on the mRNA. Well, then what you gotta do as you go from mRNA to proteins is the protein synthesis, which is also called deletion. So the ribosome is gonna read the messenger RNA, and the transfer RNA is gonna bring in the correct amino acid to the right site. Those amino acids are gonna connect together to make the protein. That's how transcription, translation work to get you to protein. The protein is just a long chain of amino acids, and then those proteins lead to the different characteristics or traits. And the tRNA is going to keep bringing those in, as you can see. So remember, what happens is the DNA stuck in the nucleus, but you make a copy of that blueprint. As you bring it out, it's like a record of your school grade, so it's like a transcript. We call it process transcription. And since you know some of these abbreviations on your transcript don't make sense, like I took biology MO1, what does that mean to someone at UCLA? So what you have to do is you have to have those secret codes translated, so then the ribosome can make the correct sequence of amino acids that are brought by the tRNAs. Those tRNAs have anticodons, which match the codons on the mRNA. And it keeps going and keeps going. Remember, those proteins are really long sequences of amino acids. Once you get that together, you get a protein. It becomes a globular protein that leads to a trait. Woo! That's biochemistry in a nutshell. And here it is in one graphic. There's your DNA, there's your transcription, your translation, and finally you have the protein, which leads to the characteristics that you're interested in. All righty. So, biochemistry. I hope this made sense to you. If it doesn't, go back, take a look at it, compare it to what you read, and I bet you'll have a much deeper understanding of biochemistry as we go along. Remember, there's a Quizlet set that you can study on biochemistry and biotechnology, along with the Quizlet set for exam 2. Study both of those and you'll do great, I'm sure. Okay, have a great day. We'll see you soon.